Are we on now?
Yes.
Oh, now we are on. Yeah. So when you see somebody with a title like this, it could mean one of two things. It could mean that they really think they have something profound to say, or it could mean that they had to provide a title and they had no idea what they were going to talk about. And so you can sort of assess that as I go forward and see where we are. Also, I'm not going to talk about any introduction. If you're here this far in this meeting and you need introduction into longevity, that's your problem, because I don't have that much time. And so I do want to point out a couple things about interventions, and I'm going to talk about biomarkers. And so I think one of the big things we have in the field is we've made huge progress in these vertical spaces. You know, we've come up with many, many different kinds of interventions that might slow aging, and you've heard about tons of those. There are many diagnostics or biologic aging clocks or other kinds of measures of aging that have made really revolutionary progress in the field, and you've heard about those. There are aging pathways, which you could call hallmarks or pillars of aging, and there's a lot of progress there as well. And then there are outcomes, which are age-related diseases or age-related cognitive decline or physical decline or whatever you want to measure with regard to aging. I think that one of the challenges we have, though, especially in humans, is that we haven't yet figured out very good ways to link all these things together. So what we really want to know, I think, is whether an intervention like alpha-ketoglutarate, where is it going to be impactful in humans in all of these verticals? Which diagnostics is it going to impact the most? Because all these biologic aging clocks are measuring overlapping but somewhat different things. Which aging pathways are the most impacted? What are the outcomes that are most relevant to alpha-ketoglutarate? And I'm only using AKG as an example. I could put a lot of different things up there. And of course, whether it affects longevity. And I think this is critical for two things. One is, we really want to be able to personalize interventions at the end of the day. And I don't think any particular intervention is going to work in everyone. And we need to know how to, at baseline, figure out what to give what people. And the other is we need to figure out how to combine things together. And so far, at least I'm very bad at doing that because we've tried a lot of combinations in mice and we're not that successful. Some things work, but a lot of things don't work or cancel each other out. And I also want to point out that I think that there are a lot of longevity clinics, some of them are here, that are doing things that are beyond what we can do in the academic sector. And it's true that a lot of these things aren't validated completely yet, but I've sort of moved to the approach that we really want to work with these clinics, assess data, help them with interventions, and try to really get a good statistical parameters on what's working and what's not. And one way to look at this is that we may not need yeast and mice and flies and worms anymore because billionaires are the new model organism. And so we should take advantage of that data. So I want to talk about one intervention, maybe briefly two, but mostly one. Just to illustrate a point, I think, about these interventions. And that's Urolithin A. And this was originally proposed by Johan Oer. And many other labs have worked on this, and it's one of the interventions that we've done a lot of testing on. This is from an earlier review showing that people eat lots of things that can be converted to urolithin A, differentially depending on who we're talking about in the gut. And then once you have urolithin A, it has beneficial effects on a whole range of different systems. And I think one point I want to make from this is that when you have one of these longevity interventions that slows aging, you can make a slide like this. It affects muscle, it affects heart. You can do this with almost any intervention. And so I think when we're trying to connect up hallmarks to interventions, as I showed on that earlier slide, it's not enough to just say the muscle's better. We need to know why. And I think one of the key things that's lacking right now is we're not doing enough work to figure out what the real targets of these interventions are, not just what the downstream events are, because those can be very indirect. And so we've done a lot of work on looking at urolithin A targets, and I think we know what a couple of them are now. But I'm not going to talk too much about that. Anyway, urolithin looks good in reviews. And so we, a few years ago, wanted to go back and try to figure out what it's doing in the context of a variety of different species. And this is one of the things we really focus on in US is we want to, our own interventions or other people's interventions, we want to bring them into the pipeline at the right place in terms of species, go backwards for mechanism, go forwards for translation, and try to get as much data as possible. So we did that with urolithin. And these 3 slides I showed last year, so I'm going to go really fast. It extends lifespan in flies, both males and females, at different doses. It extends lifespan in killifish. We love killifish now. We're working with Kotong Wei and other collaborators to look at this. There's a really robust effect. We only could do this in males. We're going back now to look more broadly. Rapamycin also works robustly. I still don't think that's published, but if I'm wrong, I apologize. And this is a precursor to what I'm going to tell you about, that urolithin is reducing mTOR signaling. It works in mice. We do studies starting at 20 months of age looking at frailty, and frailty goes up in the control animals. Interestingly, at 2 different doses of Urolithin A, you actually reverse aspects of frailty in a 2-month window. And then at the low dose, it starts creeping back up over 8 months. At the high dose, it's really not statistically different than baseline after 8 months intervention. The mice have better muscle function. I'm just showing you latency to fall on the treadmill. And they also maintain their weight better during aging than control mice. So I showed that before. Here's a deeper dive. And this is just to confirm what other people have reported. We're certainly not the first people to show this, but we see much more robust mitochondria when we treat cells. This is in cell culture with Urolithin A. You block complex 1, the mitochondria look bad, which is red. And the urolithin-treated mitochondria are much better. There's also increased mitochondrial biomass, increased PGC1 alpha. So it's not just turnover, it's biogenesis that's happening. But this is what I wanted to show, which is the mTOR pathway. So we treat with ammonium chloride and lupeptin to stimulate autophagy. What you see is that both in the control conditions and in the stimulated conditions, either with intensity of puncta or number of puncta, we get an increase in autophagic flux when we treat with urolithin. And that's partly attributable to effects on the mTOR pathway, reduced S6 kinase phosphorylation, reduced S6 phosphorylation, also reduced AKT phosphorylation, and increased AMP kinase. So this pathway is impinging upon mTOR, and we think we're learning how that's happening now. So if you go back and look at omics or you look at specific experiments, you can see that urolithin is really targeting a lot of the same pathways that Linda talked about, mTOR, autophagy, AKT, AMP kinase, and ultimately upregulating PGC1 alpha. And the point I want to make from this is that we started working on rapamycin a long time ago, and I've been trying to not be like looking under the lamppost and looking only at rapamycin and the mTOR pathway, but every time we find an intervention that works robustly, it's impacting this pathway. And so I think we have to consider this idea now that you have elevated mTOR signaling with aging, or obesity, or various other diseases, particularly in stem cells with aging, that's driving inflammatory properties. Inflammation is feeding back on mTOR, and this is a vicious cycle. And if you're working on an intervention, even if it doesn't sound like it's impacting mTOR, you might want to look at it because somewhere along the way it's going to feed into this process, I believe. Okay, so clocks. You heard about epigenetics a lot, and probably other things as well. I'm going to talk, we think that epigenetics is great, we use it, but it's nothing special about epigenetics. We can build clocks from all kinds of different complex data sets if you have the right cohort of individuals. And so we're building clocks from all kinds of things. We have clocks from lipidomics that Max is working on. We have clocks from other proteomics. But today I'm going to tell you about a clock from clinical parameters because we wanted to build something actionable that clinicians can use when they're talking about aging and longevity. And I'll just briefly touch on a metabolomic clock in the last few minutes. So this is really work of Jan Gruber, who was here and gave a talk at the Physics and Aging conference. I really want to emphasize the Physics and Aging Day. I think that we need to bring physicists into this field, and we're planning to have a whole conference on that in Singapore in the near future, so if you're interested. Jan's a physicist by training, and Feng Sheng is a geriatrician who's really brilliant, and so they put together this clock, and so the idea was take the parameters from NHANES. These are things that clinicians understand. Medical records, questionnaires, clinical laboratories, LDL, glucose, high blood pressure, 170 different features in that, and then this tried to just build an aging clock from that using a principal component strategy. And you get something that looks like an aging clock. Here's chronologic age and PC age, or biologic age. And we built this in 1 year of NHANES to predict mortality because there's 200 months of survival data for these individuals. So we can predict mortality, not age, and then work backwards to impute age. And so does it work? So we tested it in the next year's input in NHANES. So this is the test case, and it looks good. We took individuals in this 65 to 75 range and looked at their survival. So the people that, this is the survival of the whole group over 200 months. This is the ASCVD score here, the cardiovascular disease score in the red. It predicts the top 25% or do better. The lower 25% have higher mortality, not surprising. But we can do better than that clock, probably because we're looking at more aspects of aging. We're better at predicting the people that are struggling and much better at predicting the people that are going to survive at 200 months. And since we did this by principal components, we can look at those principal components, and they're all measures that you've seen before. So I just plotted it on an axis here where we have multiple dimensions. And so this group here, these are clusters of people that are the healthy people. These are the people that are going to live a long time. But then you can wander off in different trajectories from health. You can head down a cardiometabolic disease axis, and we know this because the principal components that are driving the biologic age in these individuals are measuring metabolic markers mostly. You can head down a frailty axis without a metabolic disease. You can get specific diseases, cancers, and things like that. But one thing that we were curious about, would any of the principal components just measure the distance from health, not the direction? So it doesn't matter if you're going this way or this way, it's how far you are from the green space. And there was a component that did that, and it comes back to something we've heard a lot about this morning, and that's inflammation. The component that's really driving movement away from health is inflammatory. And you can see immune system, inflammation, iron storage, red cells, and related kidney function. So this, again, I think supports the idea that changes in the inflammatory process, the immune system, are some of the early events that are precipitating a lot of the other things that are happening during the aging process. And you know, so this clock works. We made a slightly different bespoke clock using the parameters from the calorie study, and you can see that people that are on the control group here are aging 2 years over a 2-year window, and the calorie-restricted group are not showing changes in biologic age over that 2-year window. So most of that's published. I want to go on and show you a little bit of new data from this. First of all, we can go back to NHANES now and look at different groups of people. So for instance, this is across a whole adult range, and you can see that the fraction of people that have optimal health is going down with age. That's not surprising. The fraction of people that require treatment are going up with age. These are people that have clinical markers that would indicate disease, but we know what medicines they're taking. They're not taking medicines to treat that disease. So these are poorly treated individuals. That's going up with age. And you can start plotting survival curves for these different groups. So for instance, if you take the optimal people, they live a lot longer than the people that are the normal group or this borderline group. And then you have the required treatment group that have much higher mortality. This is only 120 months in this study. So let's go back and look at the clock and see how that performs. So this is biologic age in the PC4 axis. This is the aging axis, okay, not the whole clock. And this is the survival I showed you. So if you're borderline, you have a higher biologic age, a higher PC4 age. If you're optimal, you're biologically younger in the clock. That makes sense. But the surprising thing, and maybe it's not so surprising because it's been seen sort of before, is that we can look at people that have optimal markers but are taking drugs. So these are people that have been diagnosed with something, high blood pressure, high cholesterol, and they've been given drugs for it, and the drugs are working. They're optimally medicated. They're in a very good, healthy range. And the interesting thing is that they're biologically younger than the people that are considered healthy. And we can talk about what that means in a second. Let me just show you what it looks like on the survival curve. Here are the people that are healthy that are taking no drugs. And here are the people that we call polypill that are optimized for their medications. So what does that mean? So the trivial explanation, which we don't think is true, we're trying to model this to prove it, but we're not quite there yet, is that when you get diagnosed with a disease, you go to the doctor more often, you catch other conditions. Maybe a Smythe effect, but that's, I think, not the main story. I think there's two possibilities here. One is that we're not being aggressive enough with medication. And so when someone comes in and we say, oh, you don't have any clinical parameter that triggers a disease indication, we're not going to give you any drugs, a lot of those parameters are not quite healthy, but they're also not disease. And so maybe that means we need to be more aggressive about intervening. It could be lifestyle intervention, doesn't have to be drugs, but we want to get people into the optimal range. The other possibility is that these drugs, all of them, metformin, all of them, are measuring things where you just diverge a little bit from health. You're moving out of the healthy range. And so, in other words, the network is starting to fragment a little bit. And this is the trigger. And so if you hit that component, you can not only protect from the disease it's designed for, but you reestablish the network and protect from a whole range of other diseases. So I think beta blockers, statins, metformin, all of these drugs may be doing that in the right individuals. And so that's the main point from that. So I want to just end on one more clock, and that's a metabolomic clock in mice that we helped build. This is work from Peter Mullen, who's somewhere, if he didn't drink too much last night. No, he made it. He's back in the back. And Stephen Pilley, and two people from my lab, Max Unfried, who's gotten so popular now that he's artistically rendered. I don't even need to use a picture. And Joachim in my lab. And so what we did is they did all the work. I'll show you a little bit of their work, and we did a little bit of work. So they built a metabolic axis of mouse aging, taking mice from different points in time, and a whole range of different tissues, plasma, bladder, you can read them all here, and measuring how metabolites change over that window. It's a really beautiful compendium of data. And then you can take all the mice and line them up against each other independent of age and see how things correspond. And what, of course, for instance, bladder corresponds well to bladder, and that goes all the way down the group. But one thing you see is that thymus doesn't correspond well to thymus, and this is because the young and old animals are put together here, and what you're seeing is a decline in thymus function early in aging. So that we expected that, thymic involution, but at least it's good to see. Thymus and spleen are highly correlated with each other, and there's some other interesting correlations here that I don't have time to talk about. So this is what I wanted to talk about, though. We built a clock out of this, and it's not a huge data set, so it's not the best clock in the world, but we took the mice at different time points, and we tried to see, this is only plasma, although we've done it in other tissues as well, and we wanted to say, this build a clock that predicts age. So here's the test group, or here's the training group, and here's the test group. It works, my pointer's going away, but it works quite well. And then there's lots of metabolites that go into that prediction. But then we said, what's the minimum number of metabolites that gives us a high predictivity? Because the one thing I want to use with clocks is I want to get to mechanism. And so when we're working with metabolites or proteins, and we can find 8 metabolites or 8 proteins that give you 90% of the prediction, those metabolites might be highly relevant to aging. And what was encouraging to see is that one of them that came out in plasma is alpha-ketoglutarate, which is one of my favorite molecules. We've shown a lot of data on AKG. And in looking in more detail, you can see that in plasma, AKG is going down pretty dramatically with age in these animals. And I think I'm just showing this not to get into a long discussion about AKG today, happy to answer questions, but to make the point that these clocks have value beyond predicting age. We need to be using them to work backwards to mechanism so that we can understand the biology of aging. And when you're working with lipidomic clocks or metabolomic clocks or proteomic clocks, that's easier because these are actionable molecules that are doing something. So I want to encourage people to try to go backwards to mechanism from these clocks as well. So just one more, I can't even move the slides, no. Okay, good idea, Morten. One more point I want to make, and that's that I think that I'm pretty confident that we're going to go from the top to the bottom here. We've got a lot of preclinical research. Clinical studies are going on now, very exciting. Community-based studies are going on. Community-based data is becoming available. I think we're going to take health span and lifespan out a little bit in the near future. Maybe it's 5 years, maybe it's 10 years. But I don't think that any of the particular interventions, except maybe reprogramming, are likely to take us way out. So the question is, can we get from there to here? And I think that the interventions, and we talk a lot to Peter Fedichev at Gero about this, who's stuck in Singapore right now, and I think that the interventions that take us from the green to the orange may be entirely different than the ones that give us the modest benefits that we're working on right now. And what this is a plea to fund discovery science. Everything has gotten so translational. That's great. I've been in this field a long time. I love that. But we can't forget doing the basic science, because I think the big discoveries, the big questions haven't even been answered. Can we dramatically slow aging? Can we stop aging? Can we reverse aging? I think we all have maybe different opinions about that, and that's reasonable because we don't have the data to prove it one way or the other, and we need to do the basic science to answer those questions still. So please, we need to focus on both the translation and the basics. So that's all I'm going to say today. Sorry, I went really fast. I just want to highlight that Andrea Meyer's doing a lot of great clinical studies at NUS. I talked about people I think that were involved in the projects along the way, and thanks a lot. Happy to take questions.
All right. Okay. Okay. Sorry, Boris. Yeah.
Brian, terrific talk. Really great. Two small comments and a question. The first comment is about the model organism billionaires.
Sure. Yeah.
My major fear is that they die and it will reflect on all of us, right? They said if they died, what happened to them?
Yeah, I think it's a concern.
I want to ask.
Yeah, I think that,
that's not the
clinics
question,
are doing things anyway. And
Yeah.
so the question is, do we interact with them, or do we just say, oh, they shouldn't be doing that? And I think that's something we could talk about.
I like the fact that you take in hands and you're agnostic to diseases, really, because whichever disease, it came from aging.
Yeah.
But this is the question. I'm surprised the Urolithin A is so much of an mTOR inhibitor, because people say the muscle is doing great.
Yeah.
But if you take and do combination of drugs, and so many of them affect mTOR, is there an mTOR that's going to be too low?
Yeah, it could be. And I think that's some of the reason we see counteractive effects sometimes when we combine things together. By the way, rapamycin, at least in our mouse models, does not adversely affect muscle function with age. It's preserving stem cell function in muscles, and actually the mice are, by most measures, are doing at least as well as the control animals with age. So I'm not sure that's something to really worry about either. But I think that, I think that that's part of the problem, and Linda alluded to this. When we combine things together, are we hitting the same pathway? Are we hitting it better or too hard? Are we hitting combined pathways that work together? And I don't think, we don't have the rules for this right now. And so when people are out there combining multiple things together, I say this all the time, if you're taking 20 different things, it's like mixing 20 colors of paint. At the end of the day, you're going to get some ugly brown outcome, probably. And so really have to be careful with that, because I can't tell you what 3 things to take together. So just that's my opinion on it. Yeah. Yeah.
And I'm fascinated by your question about different science in order to increase maximal lifespan rather than median lifespan.
Yeah.
Did I get it right?
Yeah.
And when you think about the human body, we have over, our life, some accumulation that makes us different at age 20 from 70. And it's likely part of that is, hormonal and immune, but part of that will be cellular, the cells that remain in our body, like neurons and stem cells.
Yeah.
That means if we don't understand single-cell organism aging, we're not going to be able to understand part of the problem. And that means that we need an effort to understand even yeast, like you're an expert about yeast.
I have no argument with this.
Yeah. Which is, the tractable place to look with computation and experiment. I just want to make the comment that without that understanding single cell maximum lifespan extension, we won't get to first base.
I think it's true, and I made the comment this morning talking to someone that, we talk about systems biology, and I really don't think we've really done that. And you can do it in maybe a mammalian cell, but that's not an organism. I think there's a huge value to doing that in a bacteria or yeast cell, where there is a chance now, I think, with the data we have, to really look at things from a truly systems perspective. So yeast scientists I tell people in my lab, don't just do yeast, because I don't know how you're going to get funded to do that when you have a lab. But it's a shame because we really need to focus on these basic model organisms. They've got us a long way. There's no reason they won't take us further. So.
All right. Thank you so much,
Thanks.
Brian. We have to stop here in the interest of time, but thank you so much.